Okay, hello everybody, welcome. My name is Peter Cattini. I'm a professor in the Department of Physiology in the Faculty of Medicine at the University of Manitoba, and it's my great pleasure to facilitate this Café Scientifique, Nature and Nurture, the New Science of Epigenetics. Now, for those of you who aren't familiar with the concept of Café Scientifique, it is very much an informal discussion around scientific areas of interest. In this case, it's a health-related one, I suppose, and it's really a discussion. It is not intended to be a lecture, so it's very much audience participation. So questions, comments, everything like that. This Café Scientifique is the University of Manitoba CIHR Café Scientifique, and as the name suggests, it's funded, co-funded by the University of Manitoba through the Vice President Research and International Office and through the CIHR. And again, if you don't know who the CIHR is, then it is the largest health research funding agency in Canada, and it's really funded by you, you taxpayers and the rest of us, I suppose, that pay taxes. So, very important. In terms of the topic tonight, Nature and Nurture, the New Science of Epigenetics, I suppose if we went back many years, at least from a scientific point of view, many of us would have been working with the assumption that everything is pretty much dictated by our DNA, the genes, in other words, our genetic complement, what our parents bequeathed us. We're similar to our parents, but not the same. We have our own genetic complement. And the idea, I suppose, went along the lines that your life, or your, I suppose, risk for disease, maybe your longevity, was dictated through nature, through your genetic material. More recently, but always, there's been the view that nature wasn't all that was concerned, that nurture was a part of this. Your experience, your environment. And I suppose one of the classic examples is our friends over in Japan, who for a long time enjoyed good health with regard to heart disease. They certainly didn't enjoy the high level of heart disease we have here in North America. The interesting thing, of course, is if they emigrated to North America, that within a few generations they were enjoying the same levels of heart disease as the rest of us. So clearly something was going on there. Experience, as I say, and environment. Now, what are the implications of this? Well, this is now the subject of intense research scrutiny, and to help us in this topic tonight, we have three experts who I will introduce formally in a moment, to help us navigate through what these consequences might be and to ask questions, because really it is rather empowering to think that we ourselves can influence our health. But there are interesting issues that arise out of that, so hopefully that will frame a lot of the discussion tonight. Now I've got, before we go on to the introductions and the event prop, I've got a few bits of information. First of all, apparently, and I never thought I would say this, you can follow tonight's conversation on Twitter using the hashtag Café Scientifique, that's in one word. And in addition, we're all being filmed. And in fact, so if you enjoyed this experience so much, you can experience it again, or you can pass it on to your friends or family, or I suppose your worst enemy, come to that. And the way to do that is to visit the University of Manitoba website. That's umanitoba.ca/research/Café_Scientifique. I'm sure you all remember that, wrote that down, but you can certainly ask me about it afterwards if you need to. A few rules about this process. As I said, in a moment I'm going to introduce each speaker. They're going to speak to you for about 5 to 10 minutes. And for the speakers, at 9 minutes you'll see me do this and say, wrap it up. I'm not being rude, but we really do want the audience to have their fair share of the time. When I come round to you, if you have a question, just do so by raising your hand, and I will come up to you and hold the... this microphone has to be used in a certain way, so I will be holding it up for you. So just raise your hand and we'll try and see how that goes. If we go the full time, the last question will usually happen around 8:25. Now I should point out, if you haven't seen them, there are some refreshments on the table over there. There's also a sign-up sheet. And the value of that is that you get to hear about other events put on by the University of Manitoba, not just cafes, but visionary conversations and things like that. So please sign up if you're interested in those events. And I'll also draw your attention at this time to the evaluation sheets that you have, that you're either sitting on or on the table in front of you. Please fill these out. They are very, very useful. We need them in terms of getting funding for these kinds of events, and they also have the potential to give us some ideas about the sorts of things you'd like to see discussed at these sorts of events. So feel free to write down any topic that you have in mind, not just health related, anything that you've read about, seen, and you feel would be interesting to evaluate in this kind of forum. The last point really is an important one, and that is, as I said, this is a discussion forum, and so in terms of the questions you ask, I would encourage you not to sort of get into any issues, if you can, of specifics about treatments or things like that peculiar to you. If you can, or you have that kind of interest, try and generalize them. If you generalize them, it's fair game. And also to the panel, if you do receive a question like that, we would like to try and answer them, but if you can, try and generalize those things. So with that in mind, I'll just introduce our distinguished panel, one by one, and then each of them will speak. So on that guy over there, that's Dr. Jim Davie. He's presently the Scientific Director of the Manitoba Health Research Council, leader of the Terry Fox Research International Prairie Node, and a professor in the Department of Biochemistry and Medical Genetics at the University of Manitoba. He serves on several editorial boards of journals publishing in biochemistry, cell biology and molecular biology, and is the editor of the journal Biochemistry and Cell Biology. He has served as chair and panel member on a number of health research funding agencies, including the CIHR, and also related to cancer research as well. His research interests include, and this is a good one, epigenetic regulation of gene expression, most appropriate. He currently holds a Canada Research Chair in Chromatin Dynamics, Tier 1, and that's a pretty a small group of individuals that hold those across the country, and is past president of the Canadian Society for Molecular Biosciences. Dr. Davie received his Bachelor's of Science and PhD degrees from the University of British Columbia. His postdoctoral training in the area of chromatin structure and function was done in the laboratory of Dr. Ken Van Hold at Oregon State University. Dr. Jim Davie.
Well, thank you, Peter. Great audience tonight. So I've been in the epigenetics research field for more than 30 years now, even before it was called epigenetics. Epigenetics is actually a rather new field, only about 20 years young. So what is epigenetics? I'm sure a lot of you in the audience want to know what that is. And so I'll give you an example. Like every cell in your body has the same DNA. But do you know that a heart cell, a liver cell, a neuron, they're all very different. And what makes them different is the genes that are either turned on or the genes that are turned off. And that process is epigenetics. So that's the heart of epigenetics, is turning genes on or gene off, even though the DNA is exactly the same. So I've grown up with epigenetics, and the field has just blossomed. The amount of information that we get, it's really challenging actually to keep up with it, all the new discoveries are made day by day. But there's some little stories I want to tell you that's really, really stuck with me, because I found them so remarkable. And I'm going to tell you a few of these. The first one is stories that really reflect just how responsive the epigenetic process is. So the first one I'll give you is diet, how diet affects epigenetic program. So you've heard the phrase, you are what you eat. Well, I'll give you an example how true that is. The story I heard was with honeybees. So in the beehive, the larvae for the worker bees are just given the run-of-the-mill beehive food, but the queen-to-be is actually given the royal jelly. That's all the difference is. So one's given royal jelly and the other one's given the run-of-the-mill beehive food. And so by changing the diet, you create a queen as opposed to the worker bee. It's just the diet, and that's epigenetics at its heart. But what's happening there is there's this tiny molecule called a methyl group. And all it is a carbon and 3 hydrogens. But that little molecule, by landing in specific sites on the DNA, can turn genes on or turn genes off. It's a master switch. And this is what actually happened by feeding the larvae to the queen-to-be, the royal jelly. She actually had a change in the methyl group that, by a certain specific location, switched on genes that weren't switched on to the worker bee that ends up being sterile while she's fertile and gives rise to other bees in the hive. Another example was that really struck me was work that was done in this northern community in Sweden called Norrbotten, or the people in there, they probably were forgotten. But this is a remote area in Sweden, and they're pretty isolated, and so they had to really depend on their crops. So if it was a great harvest, they would eat well. Actually, a lot of them would eat too well. And then there was cases where the harvest was really poor, and they had famine and starvation. But when they studied the kids and the kids' kids, they realized that there was a consequence of this famine or feast with the longevity of the kids. So with those that experience the excessive food, the gluttony, if you will, their kids actually live longer, and their live shorter, and their kids live short, about 6 years. The ones that actually experienced the famine, they had longer lifespans, about 6 years. So it really is quite remarkable about the events that occur by diet and how it affects different generations. So I have a colleague, in Oregon State University who's been looking at the responsiveness of the epigenetic program with diet, although epigenetic events can change by stress, they can change by, as I say, diet, they can change by the environmental toxins and things like that. But what he was interested in is the diet, and so this is a nice example. DEET. He hasn't got the results for DEET yet, which I'm quite interested to know about, since I spray myself for mosquitoes. But what he's found is that the environmental toxins like dioxins will actually change the epigenetic events of the parent, and then you'll see it actually in 2 generations forward. So there's changes in the epigenetic programming, which we call transgenerational events, like what happened with the individuals in Mojgan. So with the case of epigenetics, we've been starting to learn a lot more about it. We now know that it changes during your lifetime. Critical time of setting your epigenetic programming is in the womb. How many of you here have actually seen those free press articles called Damage in the Womb? How many of you read that? This is to do with fatal alcohol spectrum, FASD, right? Alcohol. So alcohol taken by the mothers by binge drinking at critical periods during the early time when she probably doesn't even know that she's pregnant causes epigenetic events that can unfortunately lead to fetal alcohol spectrum disorder. That's an epigenetic event that's changed by the conditions in the womb. We know that during that time you're very susceptible to changes. Now your epigenetic programming changes as you get older. I'm looking around the room and include myself in the group where I can say, unfortunately, your epigenetic programming as you get older is just not as good as when you're younger. So things start falling apart. Your hair goes, you get gray hair, heaven knows what else goes on, but that's epigenetic kind of losing its punch. And so it changes as we get older. We also know that epigenetic events change with disease. Under like heart disease, as Peter mentioned, and cancer, and diabetes type 2. We know all of those things can change. And so as we learn more and more about epigenetics, we realize there is a lot of promise in this field. We realize that as we know more about epigenetic changes, we can use it as a diagnostic tool. For example, there's some early work now that we can look at, again, take a blood sample and look at the epigenetic signature and tell whether a child might be predisposed to type 2 diabetes. That type of research is in its infancy. We also know that epigenetics, unlike genetic events, are reversible. And in terms of clinic and how to treat individuals, that is just brilliant in the way that you can use that to generate new diagnostics and pharmaceutical drugs that can reverse epigenetic events. So you can either use it as a diagnostic to tell if you're going susceptible to a certain disease or diagnose a certain disease. But now we know that there are certain drugs that can reverse these epigenetic events to put them back on the right track. And we call those epigenetic drugs, and some of them are already in the clinic. Yeah, so the future is very bright in this field. We see that it'll tell us a lot more about ourselves. When it comes to myself, I'm sure that the whole Davie family has this habit of being late. My daughter is late all the time. I'm sure that's an epigenetic event that I got from my dad. So that's epigenetics at work there. So they think the field's quite bright, and it holds a lot of potential and promise to improve the human condition. Thank you.
Thank you, Dr. Davie. Okay, so our second expert is Dr. Mojgan Rastegar. She's an assistant professor in the regenerative medicine program and in the Department of Biochemistry and Medical Genetics at the University of Manitoba. She's currently a scientist member of the Manitoba Institute of Child Health and Molecular Genetics and Human Disease Group. She completed her PhD in biomedical sciences at the Université Catholique de Louvain in Belgium. She continued her research training in the US and Canada. In Canada, Dr. Rastegar performed her postdoctoral training at the McGill Cancer Centre in Montreal and at the Hospital for Sick Children in Toronto. Dr. Rastegar is a widely recognised researcher in the field of epigenetics in brain development and neurological disorders. She has published many research articles and review papers in recognised and high-impact journals and has been invited for seminar presentations in many national and international meetings. Dr. Rastegar has been the recipient of a dozen doctoral and postdoctoral awards, and her research is currently supported by provincial and national funding. Dr. Rastegar.
Good evening, everyone. Just to continue what Dr. Davie mentioned, epigenetics is basically how the genome is packaged. The information is kind of encoded within the packaging and tagging of the DNA and the genetic materials. The field is quite young, but we are getting a very good progress in the field. We know that epigenetics can be inherited, or by just transferring the information from the environment, it can change the life of the same generation. So some of the examples, like Dr. Davie mentioned, like the honeybee story, the nutrition will affect the readout of the same genome within the same generation, between the queen and the worker bees. Or, for example, in the case of FASD, the fetal alcohol disorder syndrome, the effect is going to pass on through the generations. This effect is not changing the genetics of the unborn child, but rather changes the marks that are tagging the genomic DNA. So another piece of information that Dr. Davie emphasizes that these marks are reversible. It's important. We can prevent or maybe come up with some sort of strategies to reverse this damage that has been done to the cells. If we know how this is basically affecting the cells, how this programming in the brain cells during development happens, perhaps we could come up with some strategies to prevent this. There are some environmental factors that we may have control over them. Maybe addiction. That is also affecting some sort of the epigenetic programming in the brain. If addiction happens during early, at young ages, then later in life you will still have those epigenetic memories in the cells. And then that may affect the children to come to suffer from those damages. For example, in some case of drug abuse, even if the drug has been removed from the system, even if there is no drug abuse anymore, the damage is already done. So the children that are going to come in the next generation may still suffer from those bad decisions that the parents or the grandparents have done already. And, so there are some other cases that we have about the environmental factors that we don't know what is the outcome. Let's say the stress that the parents are going to go through, how this would affect the children that will come. But there are some limitations when it comes to studying the human subjects. Simply, we live longer than the animals that we test in the laboratories. If you want to cover like 3 or 4 generations to see what's happening within the epigenetics and the environmental factors that affect that, you will have to have statistical analysis for like 300, 400 years. That information is not really available to us. And in some cases, there are ethical issues when it comes to studying human subjects. Or maybe there are some studies that the research from the animals that we do is not applicable to humans. So when it comes to human subjects, we do have some limitations. But still, we do have some tools in the lab. So for example, me, I'm studying neural stem cells. I use some cells that I can actually model the human disease in culture and then study what is happening in early life. One of the stuff that we study in the lab is that one protein that basically reads the DNA tags. So that carbon molecule that Dr. Davie mentioned, this protein reads that, binds to it, and basically interprets how the read of the same genomic material, what is going to be the readout. So the genomic DNA is the blueprint. The way that it's going to be expressed is what epigenetic is doing. In animal models, people have shown that cocaine or maybe FASD, alcohol exposure, alters the expression of this gene, and then that would lead to brain malfunction. So basically what we are trying to do is that to understand what is the molecular function of this protein. This protein has been discovered over 20 years, but how and it basically reads and interprets these DNA marks is still an unknown fact in the field. So basically we are trying to understand what is going on in the compromised brain and then trying to design some preventive strategies for some diseases that do not have any treatment at the moment, such as FASD or autism or Rett syndrome. Thank you.
There's a delay on. There you go. Thank you, Jim. This field, and I like this idea that it's reversible, very empowering, as I say. Is Dr. Kirk McManus, and again is assistant professor in the Department of Biochemistry and Medical Genetics at the University of Manitoba. He is also a senior scientist within the Manitoba Institute of Cell Biology, housed within Cancer Care Manitoba. He received his undergraduate bachelor's degree from the University of Manitoba and moved to Edmonton, Alberta to conduct his PhD studies in oncology under the supervision of Dr. Michael Hensel. There he studied the regulation and dynamics of post-translational histone modifications and their influence on chromosome segregation. His postdoctoral studies were performed with Dr. Phil Heiter, at the Michael Smith Laboratories in Vancouver, BC, where he utilised cross-species approaches to identify genes that regulate chromosome stability and characterise their impact on cancer development. Dr. McManus joined the University of Manitoba in June 2009, and his research interests focus on identifying and characterising genes that regulate chromosome stability in a cancer context and exploiting these characteristics to identify novel therapeutic targets. He currently couples genetics, biochemistry, and cellular biology, along with innovative digital imaging microscopy to identify both genes and targets of interest. Dr. McManus.
Thanks very much. So Jim mentioned that this is a relatively new field, but obviously it's old enough that he's referred to as my PhD grandfather, because his PhD student was my PhD supervisor. So small world. Anyways, I really became interested in epigenetics when I began my PhD work in about 1999. So I've been in the field perhaps not as long, but quite a while anyways. And really my interests go beyond sort of what's been mentioned before as far as tagging the genome or tagging parts of the DNA with certain methyl marks, and really begins to look at the second level of epigenetic regulation, which is really what we refer to as post-translational histone modifications. So if you imagine the tags on the DNA as basically on-off switches, what the epigenetic regulation of these post-translational histone modifications really works as a dimmer switch, turning up or turning down gene expression. It can turn it all the way up and on, or it can turn it all the way down and off, or somewhere in between. So it's sort of like fine-tuning gene expression. So the histones themselves are proteins, and their primary role in the cell, as in the nucleus, is to basically package the DNA. And within any one of your cells within your human body, if you took the DNA out and stretched it out in a long fiber end to end for all the chromosomes, you'd have approximately 2 meters of DNA. So that's about 6 feet in every cell. That's a huge amount of DNA that fits in a nucleus. And to kind of give you an analogy, that's like basically taking a very thin piece of thread that's about 2 miles long and throwing it into something the size of a tennis ball. So in cells, whether it be a cell from my toe, a cell from my eye, or a cell from my ear, all of that DNA is the same, just as Jim has indicated. But what is important is that at certain times during development, at certain times to differentiate certain types of tissues, or in responses to certain environmental stimuli, certain genes have to be turned on. So the DNA structure has to change so that certain transcription factories or certain elements that help express proteins can be found, and those proteins can be expressed. Conversely, at certain times you definitely don't want certain expression profiles, and you want those genes to be turned off. So the histones basically package or fold that DNA back up. And what you can imagine is those histones are just almost like a spool of thread. And the thread is the DNA, and it gets wrapped around that spool. However, unlike that spool of thread, you don't have a lot of thread going around it. Each piece of DNA goes around about 1.5 to 2 times. And that's so that those histones can slide a bit to make that DNA accessible or inaccessible to some of those factories. Or alternatively, it can be modified, such as those post-translational histone modifications that I was talking about. And the interesting thing about these modifications, in my mind, is they are a lot more complicated and a lot more complex than the DNA methylation that was referred to before. There's at least 16 different types of modifications that can occur. There's methylation that can occur, there's also acetylation and phosphorylations. And these are all unique covalent modifications or unique chemical bonds that are formed with these particular histones, or those packaging molecules, that will render the DNA accessible or inaccessible to certain factors. So really it sets up a barrier to the DNA, either making it accessible or inaccessible to some of these particular factors. And there's additional layers of complexity that are superimposed onto that, in that histones can also be methylated. But rather than just having one methyl moiety put on to some of those particular proteins, like is the case for DNA, you can actually have 1, 2, or 3 chemical additions to the same amino acid or building block within your protein, which adds a huge level of complexity. And there's very different functional outcomes associated with whether or not you have 1 of those tags or whether or not you have 3 of those particular tags. And those tags, as has already been implicated, at least for the DNA, are impacted by a variety of different factors. It can be environmental stresses. Perhaps it's brutally cold out, nobody likes to get all their coats on, and they're putting their coats on and they're stressing out just because they have to go out that door in the cold weather. That can definitely impact your epigenetics and change your expression profiles. as well, the foods that we eat. It's now spawning a huge industry that's collectively referred to as nutrigenomics. And there's a lot of research going on as far as changing your dietary component to upregulate certain gene expressions that are beneficial and perhaps prevent cancer, or conversely, certain genes that are turned on to give you cancer to actually turn those off and reverse those particular effects. And I think one of the excellent things that was mentioned, and one of the common themes is, because these modifications are reversible, they are very good targets for therapeutic purposes. And in fact, if you think about your genome and how we used to think about cancer as it's a genetic disease, you get a genetic defect in one of your genes and you get cancer. This suggests anyways, perhaps we have the ability to regulate that cancer and perhaps fight it by targeting some of these particular epigenetic modifications. And really, any of those particular modifications, whether it be the DNA or whether it be the histones, really are the result of a fine balancing act. And that balancing act really it is sort of a trifecta, if you will. It's the enzymes that add on those particular modifications, the enzymes that can cleave those particular modifications, and as well the actual donors for some of those special chemical tags that get put onto the proteins or onto the DNA. So if you change that combination by adding inhibitors or activators of some of those particular enzymes, or perhaps through dietary changes or certain supplements that you take, change the actual methyl donors or those particular things, you can impact your epigenetic profile, hopefully in a beneficial way. And I think there's a lot of research going on now that's trying to investigate this and see how we can modulate these post-translational modifications to best change our epigenetic profile. Thanks.
Thank you. All right, now it's reality TV time, since we're on camera, and it's your turn. So if you have a question, just raise your hand and I will try and hurry the mic over to you. Oh, please, don't be shy. Oh, I knew that would happen. Oh my word.
Dr. Davie, you mentioned, I think it was your last example of you being late and your daughter being late, and you thought of it. And that kind of confused me a little. I think you were being serious, but I got the impression that this epigenetics are all stimulated by chemicals and things like that. But your example seems to indicate that you can also have your genetics or the epigenetics affected by learning, observing. Is that?
Well, actually, there's some excellent work that was done by a group in Toronto, Michael Meany and Mosif from McGill, where during the early informative years and the care the mother gives it, so it's sort of like in the household and the quality of the household and the environment of the household. And so in this particular case, they had two groups of mice. One is where the mother paid a lot of attention to the pups, grooming them, a lot of licking and things like that. And then there was another group of mothers that just neglected the pups. And what happened is that they mapped all the epigenetic events up into a specific part of the brain that regulates stress responses. So although I'm giving it kind of a humorous example, but there is some reality to that type of event. And then they went on further. They initially did it with animal work, and then they wondered whether the unfortunate souls that committed suicide, whether there was any epigenetic event that occurred in their lives in terms of the stress and the households they were from. And actually they did find that if the child was from a neglected family, they would see the same epigenetic events in the hippocampus of the brains of these young ladies that committed suicide. So the quality of the environment, the quality of the interaction during those informative years, yes, it has an epigenetic
consequence.
But I think just to add to Jim's comment, is much of the work that's come out of many of these animal model systems or even some of the human events has definitely impacted or sort of brought to the forefront the influence that the mother has on the epigenetic profiles, which is perhaps underappreciated at the moment is the father's input. There's not a lot of studies on that. There's a lot of information out there that perhaps says we're useless, but that's not necessarily the case. I think if we go back to Jim's initial observation that he was talking about, as far as the hardships in Scandinavia and whatnot, it was actually the father's side that actually influenced whether or not the children would live longer under starvation. And so there is definitely both a maternal component as well as paternal component. So there's a lot of research that's actively going on trying to assess what exactly those components are and how they impact your epigenetic profiles.
Next question.
Firstly, since they're reversible, and you mentioned loss of hair or the aging, now what's the chance of slowing down that process and getting that hair back?
Yeah, I'm interested in that one.
I think the proof is in the pudding. I haven't figured that one out yet.
If we did, we'd be done.
Fine balance, and the problem with epigenetics is yes, it's reversible, and yes, we can inhibit some of these enzymes that perhaps are causing the problem. But the problem is they're affecting hundreds of genes, and so you have to dissect. It's not like in the past where people were looking at genetics, they would knock out their favorite gene and they would look for an effect. The problem is if you start to toy with some of these epigenetic phenomena, they're potentially impacting hundreds of genes. One of the other things that we haven't even spoken about is many of these proteins that impact those tags not only tag the DNA, or more specifically the histones, but they also tag other non-histone proteins and have functional implications for those. So right now we're sort of trying to focus our research down on their impact on histones and gene expression, but there are a myriad of other proteins that are also being impacted that it's going to take a lot of time to decipher what exactly is going on.
Raise your hand.
My name is Ed Benjamin. It seems to me that maybe just as human beings we're always trying to change some aspect of our physical or mental lives. How can a layman know that what he's trying to change is epigenetic and it's worth going through a lot of stress and effort to make changes because it'll pay off, versus it's genetic? So the best thing you could possibly do is just accept it and relax and realize that that's just the way that it is. How does one, as a layman, figure out which one is which?
Mojgan?
Yeah.
So the best example that some of the scientists at the University of Manitoba are trying to address is that they're looking at the identical twins. In that case, the genomic material is the same, but in some cases one of the two twins has autism, the other one doesn't. So what is your explanation for that? It must be epigenetics because the genetics is the same. So that's looking into identical twins is now the subject of many labs to tease out the impact of epigenetics on different kind of diseases.
Jim?
Yeah, I mean, your life expectancy is by your genetics. You can stretch that out to the max by manipulating your epigenetics. So a healthy diet. I mean, you've heard this about ways to prevent cancer and things like that, a healthy lifestyle, avoiding excessive drinking, avoiding smoking, and all those things will actually allow you to keep a robust epigenetic program that will maximize your genetic, whatever you're dealt with.
So just to add up into that, your genes are what your genes are, but your epigenetics has the ability to impact that to a certain extent. And just to give you an example, as far as some of the inherited cancers are concerned, a certain type of colorectal cancer, if you inherit defects in some of these genes, it doesn't mean you absolutely get that particular cancer. In fact, in some instances, you have about an 80% chance of developing that particular form of familial colorectal cancer. So it means that there's 20% that don't get this particular cancer. Why is that? Well, I mean, it's not clear exactly at this moment, but perhaps it has something to do with the epigenetics in those particular individuals. However, there are also inherited forms of colorectal cancer where if you inherit those specific genetic defects, you have a 100% chance of developing that disease as well. So I think your genetics are your genetics. You inherit that. But superimposed on top of that is the regulation that comes through the epigenetics. Sometimes that can work in your favor, and sometimes that can't work in your favor. So how exactly can you modify that in a beneficial way? Fantastic question, and I think we would all answer, we don't know at this particular point. Again, it's a very young field, and we're really trying to find out what's going on.
Dr. Rastegar, you mentioned the first time autism in the same sentence almost that you mentioned people with fetal alcohol syndrome. Now, if you have this type of phenomenon happening, and a lot of it is in womb, and mind you, some children don't get autism until they're 5, 6, 7 years old, and obviously you're saying that some genetic thing kicks in, and this epigenetics starts that ball rolling. And there's other things, whole divisions of people living in certain areas becoming similar, even in their diseases and so on. With this, my question is what are the chances of a person who is born with some form of autism, and you have all of your ones that go with that, Asperger's and so on, what are the chances that if they have children, will their children be really badly predisposed to having the same type of thing, or same with fetal alcohol syndrome, because there's a large number of fetal alcohol syndrome people being born out there and growing up into adulthood and probably getting married and so on, like anybody else. But how would we be able to stop or put a sort of a hold on that for them, so that they could be safe and feel free, to propagate?
Overall, autism is a very complex disorder, and there are some genetics and epigenetics involved in autism. So in cases of genetics, if the cause of autism is genetics, it could well be passed on to the other generation. But in the brain of autistic patients, when people looked at the gene that we are studying, MECP2, it's a reader of the DNA. The expression of this gene in the brain has been reduced. So the expression is quite critical. Even twofold less or twofold more expression causes intellectual disabilities. In the cases that it's epigenetics, I'm not sure how well that would be passed on to the other generations. Same thing for FASD. I'm not sure how, it is epigenetics, FASD. You may find some genes as markers that the expression has been changed, but how well that would be passed on, I'm not sure. Jim, would you want to comment on the transgeneration? And
yeah, well, there's critical times during early embryogenesis and in the first trimester particularly, where there is the ability to change some of the methyl marks that we talked about on the DNA. And that's thought to be at the heart of transgenerational. So there's a lot of work still to be done to really fully understand how those events occur, and how the signature on the DNA with the methylation has gone and passed on from one generation to another, and what's the mechanism of that. There's a lot of research on there. We just don't have the answers right now.
Okay, next question.
I heard during this discussion there was a bit of mention of companies now that were starting up that were trying to sort of market the idea of knowing your epigenetics or shifting them, and I'm kind of wondering at this stage, are they kind of more on the territory of sort of, I suppose, snake oil salesmen, or are they, do they have more of a legitimate basis, and if so, are we going to see like a transition from more the snake oil salesman to the legitimate basis and having a real impact on people in the future, or where is this sort of currently?
So perhaps I'll start with this one. Personally, I would argue that they're more the way of snake oil right now, because we just don't really have a good appreciation as to how changing your diet is really going to impact the hundreds of genes. In certain instances, it works wonderfully. In certain instances, there are definitely therapeutics that are targeting some of these enzymes that add those modifications or remove those particular modifications. And in certain cancer contexts, they've been proven to be very effective. In fact, some of these chemicals have gone through initial preclinical work into clinical studies, and some of them have actually been approved by the Federal Drug Administration in the United States. Other ones, of course, have failed and gone to the wayside. But I don't think we appreciate enough at the moment exactly how some of these particular drugs or compounds are changing our entire epigenetic profile. We're looking at a few genes generally. We have next generation sequencing technologies that are helping understand how these changes are applying to the genome. But again, there's a lot more complexity that we're just not understanding, and some of these particular modifications or chemicals are also going to affect non-histone proteins or non-epigenetic phenomena as well, and we just don't have a good handle on them. So in certain cellular contexts, they work well. And in another one, they may not work well at all and actually may be quite poor choice to take. So it's a balancing act at the moment, and we just don't have a clear answer.
I just had a question about the bee example. So what I got from that is that they fed like the, is there a larva stage or something?
Yeah, that's right.
Of the bees, and then the one they fed the extra good stuff turned into queen bees.
No, this is actually what actually happens in the hive. Is the queen bee will be fed the royal jelly, so they're pretty selective. The one when it's time to have a queen, it's selected to have the royal jelly. So most of the time, the worker bees, they just get the...
So how do they decide, like, which one becomes the queen?
That's not what you're reading. But imagine that as the queen gets older, it's...
None of us were the chosen ones.
Rushing around here to the front, sorry.
I wonder if any new light is being shed on Alzheimer's disease in relation to genes being turned on or off.
I think Mojgan first on that. Is that okay?
I'm not sure. My field is more towards the autism and Rett syndrome. I don't work with Alzheimer's. But there are many groups that basically work with the, right now there is a lot of stem cell modeling of different diseases that are happening. So basically in cases of neuronal disorders, for example, it's very hard to get the neurons and then study these neurons. There has been one breakthrough, basically Shinya Yamanaka from Japan that got the Nobel Prize also. Last year, he generated some reprogramming epigenetic. He started with adding some genetic cocktail to the cells, the normal cells, and then reprogrammed them back in time into early developmental stage. And then at that point, you can start manipulating these cells back to any kind of cells that you want, like nerve cells, muscle cells, heart cells, anything that you want to study. And then at that point, you can get some cells from a patient that has Alzheimer's, make those cells, and then now make them to neurons, and then you can study what's happening in terms of genetics, epigenetics. But basically, my field is not Alzheimer's. I'm sorry. I don't know.
Right.
And so any of these events could have led to a change in epigenetic processes of specific neuronal regions of the brain that affect their mental being.
I mean, as far as schizophrenia is concerned, for at least 30 years they've been prescribing valproic acid, which is an HDAC inhibitor, as a mood stabilizer. And only more recently have they come to appreciate that it is an HDAC inhibitor and that it's actually changing the expression profile of multiple genes, hundreds of genes. So what exactly is the smoking gun? It's currently unknown.
And I've certainly seen studies being done with regard to music and looking for effects, epigenetic effects, just from different types of music and playing an instrument, playing music. That's being looked at. So these things are being pursued. Next question.
How do you explain the difference between familial and hereditary?
Familial cancers are hereditary. To me, they're part and parcel. It's basically.
It doesn't matter what disease or condition.
I always think of things in terms of cancer because that's my research area. So as far as familial, that's another way for us to say it's an inherited form. You basically inherit certain genetic defects. You don't inherit a cancer, you inherit a predisposition to develop a cancer if conditions are right or wrong.
Sorry. And walked away from you.
Wow. So they're basically interchangeable expressions.
For my context there.
Any dissension in that with our panel?
Not really. I mean, when you are actually, this is where your genetic, whatever you're dealt with in your DNA, has pretty well set the maximum that you can live and what potential, what lies awaiting for you if your epigenetic lets it loose, or if your epigenetic can't regulate it, or it regulates the epigenetic event. It can go any of those ways.
Well, one of the things building on that, one of the things that's almost an inevitability is if you live longer, you probably develop cancers. And in fact of the matter is, you start with a fair amount of methylation, DNA methylation when you're born. It actually gets erased during embryogenesis. It gets reinitiated, and then it begins to decrease with time. And so the loss of that methylation results in an increase in expression of genes that are perhaps detrimental to the cells or cancer promoting. So that's often how some of these particular predispositions can actually come to fruition with respect to a cancer.
I suppose we're all familiar with the story of the 100-year-old person being interviewed, and they asked them, and this person proudly boasts, I smoked every day of my adult life and I drank to excess, and they're smiling away and they're 100 years old, and then you hear that unfortunate story of the 30, 40-year-old individual that never smoked and really good health, and then works for 2 or 3 years in a smoke-filled bar and then gets diagnosed with inoperable lung cancer. And so you make the case that if that person never encountered that particular environment, they might have lived to be 102, but they were always predisposed to that.
So just to build on that, cigarette smoking, for every 3 cigarettes that you smoke, you generate 1 genetic mutation. Okay. But superimposed on top of that is the chemicals that you're actually inhaling, and many of those actually are modulators of your epigenetic program as well. So not only do you get a hit by the cigarette as far as your actually genetics, but it also affects your epigenetics as well.
Okay, next question.
Are there cell types in the body that are much more susceptible to epigenetic regulation compared to others?
Well, I think from a nutritional point of view, I mean, for the cells that are in the blood, the serum, since they will see, like when you're eating, it goes through the colon, go to the blood system, that would be your first hit. And then depending on, it's a concentration argument, that the higher the concentration of the agent, the time that it is circulating, what cells will actually get the compound that will be an effective dosage. Those will be all of the sort of points you have to consider in the responsiveness of any particular drug or any agent that's going to change your epigenetic profile. So you have to, it's a pharmacokinetic and availability argument. So that's how you have to answer that one.
Next question.
It raises some interesting questions in terms of the whole epigenetics as the area develops. one can ultimately get a profile, get your individual profile, and the last thing you would want to have is an insurance company getting hold of that, because they would love to find out. And so how does one ensure that that type of information remains confidential and out of the hands of these individuals and companies?
But, you know, this is, you know, I mean, for this type of epigenetic mapping, that's still far away, because you'd have to epigenetic map every cell, because the epigenetic events and signature would be different. But what you do have available now, if you want to spend a few thousand on, is mapping all your DNA, getting all your genetic code and mapping all that. And so the idea that the insurance companies would get involved in that is horrifying, actually. That they would say, well, do you have, I'm just thinking what happened to some of the people down in the States because they couldn't read the small print, right? And so you would have to get a lawyer involved if that ever came into play, and they say, well, did you ever get your genes, did you ever get your DNA sequence? What sort of genes, defects was there that might predispose you to disease? Can you imagine that? I hate to even, like if the insurance companies go there, we got problems.
Yeah, no, and I definitely agree with that as well. I think it's out of the hands of the scientists at that point, and obviously the legalities have to take over. But what I want to interject into that is, it's great to think about the insurance companies. If they get a hold of that information, what would they do with it? But what would you do with that kind of information if you saw your genetic profile and you saw all the diseases that you're predisposed for? And the reason why I bring that up is simply because there was an individual from the States, his name is Mike Snyder, who's a very well-known, very famous professor who basically monitored everything he could within his body for 2 years. The cost was over $2 million to do this. And he says one of the first things that he realized is if we're remotely anxious about anything, this is the worst possible exercise you could ever do. But it was interesting, and this is really the idea of personalized medicine, is he could monitor virtually every level in his body. His entire genome was sequenced. He could monitor his epigenetics. He could monitor various levels of sugars or whatever in his blood. And he could see changes depending on whether or not he was physically active, whether or not he was eating certain foods and things like that. So the environment definitely regulates your epigenetics quite quickly. And I mean, our genetics doesn't change all that rapidly. It takes hundreds to thousands to millions of years for species to evolve. But when you have certain stimuli, your epigenetics has the capability to respond very quickly.
I think you've got a case of too much information. If you start to think that where you live, where you literally live, is affecting you, and then you move somewhere, it's affecting you, then you start to think, well, maybe I'll just stay at home. It's got to be a box with regulated air, with a certain type of food, and it's rather debilitating in a way. It's really quite scary thought overall for them.
And maybe you don't get excitement, and
that's what, yeah.